Hello and welcome to today's webinar on almond protein extractability and utilization in a plant-forward world, presented by the Almond Board of California. I'm Swati Kalgaonkar, Associate Director, Nutrition Research Program at the Almond Board, and I'm pleased to introduce this robust panel of experts that will be providing a deep dive into plant protein using almonds as a primary example. For today's presentation, I will kick things off with a quick review of protein, complementary proteins, and protein quality, specifically the almond protein nutrition profile. Next, Dr. Juliana Bell from the University of California, Davis will present research and processes in protein extraction, including utilizing enzymes to help extract almond protein. Then Guangwei Wang from the Almond Board will take us through current almond protein technologies and the functional properties of the almond protein powders that can be ideal for solubility and digestibility. Finally, Chef Christine Farkas will join us to bring it all to life with practical and functional applications of almond protein powder in 2 culinary examples. Following all the presentations, we'll conduct a Q&A, so please feel free to drop questions along the way into the chat box, and we look forward to addressing those at the end. So without further ado, let's go ahead and get started with this webinar. So we're all familiar with the USDA MyPlate and the fact that protein is an integral part of a well-balanced diet. We're also all aware of the many important functions of protein in our bodies, everything from repair and maintenance of tissues and muscles to providing energy, transport, and storage, and working as enzymes, antibodies, and hormones. What's interesting in the world of protein is the transition that we're observing from protein that could walk on all 4 legs to protein that now grows on plants. This is where almonds fit in, and in terms of quantity of protein, they are the tree nut highest in protein at 6 grams per serving. When considering protein quality, or PDCAS values, which stands for protein digestibility corrected amino acid values, almonds are on the lower end of the spectrum, but that's primarily because they are low in lysine. As a general rule of thumb, amino acids that are most limited in plant proteins are lysine, methionine, and cysteine. Now, legumes tend to be deficient in methionine and cysteine. However, grains, nuts, and seeds that can be deficient in lysine are actually high in methionine and cysteine. So thus, there are these plant-based proteins that can really very well complement each other. The idea of using complementary proteins is not a novel one. We all do it on a daily basis as we eat diets and not single foods for specific benefits. So the idea of using protein sources that complement each other is very practical and already part of our dietary habits. Now, this slide is a great illustration of how protein quality can be improved by marrying one source of protein to another. So if you look at the y-axis here, which is where the almond protein starts out, you see that by adding different percentages of pulses, you can actually help to increase or improve the PDCAS value scores. So plant protein partnership may be an effective method for ensuring protein quality delivery. Having said that, it is important to understand that protein quality is not the only aspect of protein that one should look at. Since we eat several foods as part of our diet every day, protein partnership and hence quality can be easily achieved. But the other important aspect to keep in mind is protein quantity, and it's important for an adult to get an average of 50 grams of protein per day into their daily diets. And almonds are a great way of including 6 grams of plant protein per serving. In addition, almond nutrient profile offers a great nutrient package with one serving, which is an ounce or 28 grams, providing over 10% of the daily value for fiber and 20% of the daily value for magnesium, a nutrient that research shows is important to help reduce inflammation and inflammation-related diseases such as cardiovascular disease and type 2 diabetes. One serving of almonds also provides 50% of the daily value for vitamin E, a well-known antioxidant that fights against inflammatory agents. So almonds are a powerhouse of several beneficial nutrients. But for the sake of today's topic, it's important to state again that they are also a good way of adding plant protein to one's diet. So let's have that be the takeaway for today. So now that we've laid the groundwork, I'll pass it over to Juliana to provide a deeper dive into protein extraction. Juliana, please take it away.
Well, good morning, everyone, and thanks for the opportunity to participate in today's meeting. I'm going to talk about the development of a bio-guided extraction approach to improve the functional and biological properties of almond proteins. Protein-rich foods, especially plant-based proteins, it's a growing segment in the food industry. The increase in sales of plant-based proteins has increased the demand for using proteins as ingredients in food manufacturing. And that has prompted a significant amount of research to develop and to optimize the extraction process to produce dietary proteins with unique composition, functional, and biological properties. The development of structure function-based methods has become a key step to improve our understanding of the effects of those extraction conditions on the properties of the extracted proteins. Protein utilization requires upstream lipid removal. For example, almonds might contain up to 24% of proteins, but they might contain up to 55% of lipids. Those lipids, they are often removed prior to the extraction of the protein, and that can be accomplished either by direct solvent extraction, which is commonly used to extract soybean oil. That process is efficient and cost-effective. However, the use of hexane has raised several environmental, safety, and health concerns. On the other hand, mechanical pressing, which is the method of choice to process tree nuts, such as almonds, can also be used to produce a high-quality oil, and a protein meal which might still contain a significant amount of lipids, and that the amount of lipids will vary depending on the processing conditions used. So my research group has been working on the development of an environmentally friendly extraction approach that makes use of mechanical treatments such as flaking, grinding, extrusion, to break down the cell walls and to help to release the intracellular components, water, and enzymes to simultaneously extract lipids, proteins, and carbohydrates from several food matrices, all of that without the use of flammable solvents. The major fractions produced by that technique are a skim fraction, which is an aqueous fraction that contains the extracted protein, a cream fraction, which is an emulsion that contains the extracted lipids. So as you can see, lipids are not extracted as free oil, so that emulsion needs to be broken down to free the oil for subsequent utilization. And the last fraction is an insoluble fraction, which is a fiber-rich fraction. That fraction will contain everything that was not extracted in that process. Processing challenges associated in this strategy. To maximize extraction yields of both lipids and proteins, that's necessary to improve the process feasibility, to maximize the recovery of the extracted oil, although extracted, if we are not able to break down that emulsion to free that oil, we won't be able to produce oil for subsequent utilization. And the last one is either to maintain or to improve the functionality of the extracted protein. So adequate processing optimization can lead to the production of compounds with unique functional and biological properties. The major goal of this work was to establish a connection between extractability, recovery, and functionality. Our goal was to identify processing conditions that would lead to high extraction of lipids and proteins from almond flour, as well as to identify processing conditions that would lead to a higher recovery of that extracted oil. So as we can see in this slide, we have the solubility of proteins that were produced under different conditions. So regardless of the extraction conditions used, we can see that almond protein solubility was extremely high at alkaline pH. It varied from 98 to 99%. However, at pH 5, which is a pH where almond protein solubility is not favored, we can see that proteins that were extracted at pH 9 with the use of enzyme had a much higher solubility compared with the control where enzyme was not used. So those proteins, they would be useful for food applications involving acidic pH, including sport drinks and fruit juice. So based on those results, we did select that extraction condition involving the use of an alkaline pH and use of enzyme to scale up the process from 1 to 10 liters with the goal of producing adequate amounts of lipids and proteins for subsequent characterization and for functional studies. At pilot scale, we saw that using enzyme during the extraction led to a significant increase in oil and protein extractability. Oil extraction yields increased from 62 to 67%, and protein extraction increased from 65 to 74%. However, we can see that the extracted oil was still entrapped in that emulsion. So our next step was to evaluate different demulsification strategies to break down that emulsion and to free that oil for subsequent utilization. We have evaluated an enzymatic demulsification approach. We used the fractional factorial design where we looked at the effects of pH, temperature, time, amount of enzyme, and based on the significance of the effect observed, and also based on the type of the effect observed, if it was positive or if it was negative, we either increase that range of values evaluated or we decrease, with the goal of identifying the best conditions to break down the emulsion of the creams that were produced by the AOCS process and by the enzyme-assisted AOCS process. We did a similar approach for the chemical demulsification strategy, where we looked at the effects of temperature and time at pH 5. For the demulsification, creams that were produced from the AOCS extraction process and by the enzyme-assisted AOCS extraction process, they were subjected to different incubation conditions, which varied based on the experimental design. And after the demulsification, that mixture was centrifuged, and the amount of oil released was measured, and it was used to quantify the demulsification yield for each strategy. The enzymatic demulsification of both creams showed us that using enzyme during the extraction increased the amount of oil recovered from that emulsion from 66% to 93%. And those results are in agreement with the literature, which has shown that Often, using enzymes during the extraction does lead to the production of a weaker emulsion that can be more easily broken down and more free oil can be released. And as we can see, each cream had different optimum conditions, and that's related to the difference in the composition of those creams, since one was obtained without the use of enzyme during the extraction, and the other one was obtained with the use of enzyme during the extraction. A similar trend was observed when doing chemical demulsification. Again, by using enzyme during the extraction, we increased the recovery of the extracted oil from 68 to 81%. But as you can see, the enzymatic demulsification was more efficient than the chemical demulsification in terms of recovering more of the extracted oil. Our next step was to evaluate the effects of those extraction conditions on the protein solubility. Solubility is likely one of the most important functional properties of a protein, as a good solubility expands the potential applications of that protein. And it is a function of hydrophilicity and electrostatic repulsion. As we can see, almond protein solubility followed that traditional U-shaped curve with higher solubility at alkaline pH and low pH values. And we can see that at pH 5, where almond protein solubility is unfavored, we can see that the hydrolyzed proteins in gray, the EAP protein, had a higher solubility compared with the unhydrolyzed protein. That reflects the action of the enzyme during the extraction, which produces smaller and more soluble peptides. And that is in agreement with the zeta potential values showed in this graphic. As you can see where that red arrow is, the zeta potential of the unhydrolyzed protein is 0 around pH 5. And that is in agreement with the literature, which states that the isolectic point of almond proteins is around 5, which means there is minimum solubility of that protein at that particular pH. And at that same pH, we can see that the hydrolyzed scheme has a much higher absolute zeta potential value, which favors the solubility of that protein at pH 5. That's why we have that higher solubility at pH 5 for the hydrolyzed scheme. We also looked at the effects of those extraction conditions on the emulsification properties, on the ability of that protein to form and to stabilize emulsions. And we saw a similar trend to the one observed for solubility, with the hydrolyzed protein having a higher emulsification activity index at pH 5. However, when we looked at the stability of that protein, we saw that the hydrolysates, they had a reduced stability compared with the unhydrolyzed protein. And that's not surprising, since smaller and more soluble peptides, although they can migrate to the interface and interact with lipids, sometimes they are not able to stabilize an emulsion as well as a native protein. And those results are in agreement with the changes observed for surface hydrophobicity. We see a decrease in surface hydrophobicity when moving from unhydrolyzed to hydrolyzed proteins, which might have altered the ability to form and stabilize emulsions for the hydrolyzed protein. Another question we had in mind was if we could manipulate those processing conditions to increase the protein digestibility. And indeed, we can see that by using enzyme during the extraction, almond protein digestibility increased from 79 to 88%. And by further optimizing that hydrolysis, we were able to model processing conditions using different enzymes at different incubation times that led to a digestibility of 93%, which is quite high. So overall, the use of enzyme during the extraction not only led to the production of more soluble peptides, but also to the production of more digestible peptides. And the last question we had in mind was, can we tailor those conditions to reduce allergenicity of almond proteins. We can see that when we move from orange to blue, we are moving from unhydrolyzed to hydrolyzed proteins. There is a significant reduction in the detection of almond proteins when using an ELISA kit. However, that kit did not tell us if those hydrolysates, they could still enact an allergic reaction. So in order to better answer that question, we did partnered with immunologists, experts, and although this is a work in progress, we had some preliminary results that were very exciting. Moving from left to right, we are moving from unhydrolyzed to more hydrolyzed proteins. And we can see that there is a significant reduction in the binding of those hydrolysates, suggesting that enzyme bioprocessing did indeed reduce protein allergenicity. As right now, this is a qualitative work. We are working in the sense of providing quantitative measurements for that observe the reduction in allergenicity. In conclusions, we did identify processing conditions which involved the use of alkaline pH and use of protease that not only led to higher overall extractability of lipids and proteins, we also identified processing conditions that increased the recovery of the extracted oil by the use of an enzymatic demulsification approach. And those processing conditions, they were able to produce proteins with higher solubility at both acidic and alkaline pH, higher digestibility, and reduced protein allergenicity. The next step in this area in this work is to identify potential applications for that extracted protein, which is going to require some product development research. With that, I would like to thank you for your attention, and I would like to pass on to Guang Wei, which will review the functional properties of almond protein powders.
Good morning. Thank you to dive into our almond protein session. I want to appreciate Dr. Bear to share their latest work on the almond protein. Through their innovative research, they created 3 functional ingredients out of almond: vitamin E-rich almond oil, and mineral-rich dietary fiber, and then protein cream. I hope those can stimulate some interest from our food technologists to eventually create some commercial ingredients out of almond. But before we have those functional almond ingredients, I want to share with you what commercially available almond form being used in the plant protein-based applications. So there's several forms available being used by food technology. Some are popular a lot because of high nutrition value, but technologists use almond also complement the texture profile of a finished product. There's a whole almond, sliced almond, slivered almond, diced almond, flour, and all those in both in blanch or natural form. Natural means with the skin on. Recently, we have seen increased demand on the fine ground particles, like a flour, like a meal, and butter or paste, even put into the almond milk. So those is due to increased interest of a consumer on plant protein-based product, especially for those gluten-free applications. So all those fine ground form have in roast or raw or in blanch or natural form. So there are so many forms can be used for different needs or for different applications. So recently I have seen a great demand on the partially defat almond powder. Defatting process remove portion of fat, increase proportionally increased almond protein, mineral content, and dietary fiber. Defatting process also change functionality of a product, make a product finer, less than 100 micron in the particle measurement, and also make the powder more suspensible, so make it easier to be blend in the end formulation. So before you, the technologist, to take a Dr. Bear's approach, make functional ingredients out of almond product, we do have several commercial available product, like a fine ground almond powder, almond flour, or partial defat almond powder to be used in your pursuit for plant protein-based application. I will pass on to Chef Christine Farkas, who will showcase almond protein uses in several plant protein-based applications from her kitchen. Thank you.
Thank you so much. I'm excited to be here. Hi, everyone. My name is Christine Farkas, and I'm a Red Seal chef and the founder at iHeartFood Consulting, where I help chefs and food manufacturers across North America bring innovation to product development. I am so thrilled to wrap up our webinar today by bringing defatted almond flour, or almond protein as it's also known, to life. As you all know, consumers have an increasing desire to fuel their body with natural, transparent, and holistic ingredients. In fact, Innova Market Insights' top 10 consumer trends for 2021 includes plant-based diets, noting plant-based trends are reaching global phenomenon status. As plant-based eating continues to rise, almond protein powder shines as a clean ingredient for a variety of applications. Let's take a closer look at almond protein powder next to almond flour here. So the protein next to the almond flour, you can see how almond protein powder has a slightly different color and an extra fine texture in comparison to the flour. This is because almond protein powder is very similar to almond flour, but the difference being a percentage of the oil removed. So depending on the amount of oil removed in the protein itself, the result provides a source of protein without as many calories or fat per serving when compared to almond flour, almond meal pieces, or whole almonds themselves. When it comes to flavor, unsweetened varieties offer a clean profile that blends easily into recipes, allowing for more versatility in a range of food products and less flavor masking agents needed when developing recipes and formulations. Another benefit of almond protein is its digestibility and solubility. Because of this, almond protein not only blends smoothly into beverages and product bases without grittiness, but it also offers digestive benefits. So defatted almond flour is an ideal ingredient for adding density to bars and beverages, and can even operate as a binder in chill blended products, crusts, and baked goods. The ingredient's higher solubility enables a creamy mouthfeel without imparting an off flavor and texture in the final product. So in terms of applications, almond protein powder complements both sweet as well as savory profiles, which is ideal, while offering a light nutty note with a hint of sweetness. Here are prepared two of the many ways you can use almond protein powder to enhance nutrition and sensory profiles. First, we prepared a banana bread smoothie with almond protein and white beans. In this case, we're using whole cooked beans, but purees can also be used. This is an adaptation on a previous recipe where we used almond butter in the formulation. In this version, we are actually exploring the almond protein combined with white beans to increase the overall protein content. Almonds are high in amino acids that legumes and pulses lack, so their profiles are complementary, and by pairing both almonds with white beans in this recipe, we create a complete protein. Functionally, the defatted almond flour blends seamlessly into the smoothie without clumping, enhancing the overall mouthfeel and allows an accompanying ingredients to shine through. In this case, the banana and the warming spices of cinnamon and nutmeg, and as well as the slight nuttiness that you'd actually get from the almond milk in itself, which is also in this recipe. We're able to increase original protein level of 8 grams to 14 grams for an 8-ounce portion, reduce fat content of 10 grams to 2 grams per serving, and reduce the total calories of 270 to 200 calories per 8-ounce portion. Next, we have a press-style lemon lavender almond bar. In this recipe, we've incorporated the defatted almond flour and almond slices, along with hemp hearts, chia seeds, lavender, as well as lemon. The defatted almond flour binds with the dried fruit and seeds in this recipe, creating a clean label, no sugar added, chewy, easy to snack on the go snack. The defatted almond flour blends easily into the mixture, creating a smooth mouthfeel and a clean aftertaste. The sliced almonds create textural interest in the mixture and provides an aesthetic crunch on top as the garnish. This recipe is highly adaptable and plays well with a range of dried fruit and flavoring options. Once again, really allowing accompanying ingredients to shine through in the final product. Nutritionally speaking, for 1 bar, so roughly 52 grams, we're looking at 5 grams of fat, 10 grams of protein with a total count of 180 calories per portion. So this is just the tip of the iceberg when it comes to ideas, and there's so many opportunities when it comes to using almond protein powder in applications. I look forward to seeing the incredible innovations manufacturers and chefs will launch in 2021 and beyond. I hope you all feel confident to experiment with this ingredient in your own kitchens and for future R&D. And with that, I'll pass it back to Swati with the almond board to field any questions from the audience. Thank you so much. With the almond board to field any questions from the audience. Thank you so much.
Thank you, Christine. What a great way to incorporate almond protein into these fun and healthy recipes. So with that, I'd like to thank Juliana, Guangwei, and Christine for rounding out this review of almond protein. And there was so much great information here. And so with that, I'd like to invite the panel to answer some questions that we should be getting soon here from the audience. Again, please send through any questions you might have to the chat box. And I do see a couple of questions that are here, so maybe we'll go ahead and get started. So with that, our first question goes to Juliana. Juliana, the question is, did you compare both endo and exopeptidases as part of your enzyme-based extraction? And if so, could you speak a little bit to it, what the results looked like?
No, we did not test for that specific work. We only used an endoprotease. That's something that we are currently looking in another work, is to see, to test different additional protease. We are also interested in looking at the effect of those the use of different enzymes under different conditions on the bitterness of those components. So that's an ongoing work.
Okay, well, that's great. It's good to know that it's ongoing and something to stay tuned in for. Since I have you here, Juliana, might as well ask you a very quick question that I'm personally really curious about, which is what prompted your research into protein extractability?
First, I would say the importance of proteins for human health, but also the need of developing better and more sustainable process to extract those components. In addition to the fact that we need to increase our level of understanding of the effects of those extraction conditions on the structure of the protein and how those modifications can alter the technological properties of that protein, as well as the biological properties of the extracted protein.
Okay, thank you. I do see a lot of interest in the enzyme-based extraction here, Juliana. So a follow-up question for you is, was one of your enzymes a lipase?
No, we didn't use a lipase. We do manage to extract both proteins and lipids just with the use of a protease by hydrolyzing the proteins, extracting the protein out of the matrix that enables the oil to be washed out of the flour. So we can simultaneously extract both lipids and proteins, even though we are not using a lipase. But those proteases, they can also act on the membrane of the lipids, which help also with the extraction of the lipids.
Okay, that's great. So, and is it a very clean extraction given that you're actually extracting these two fractions at the same time?
Yes, that's true. We do extract lipids, proteins, and carbohydrates, but we do have a byproduct with the insoluble fraction, and that's also important to identify applications for that fraction to maximize the use of all fractions that are generated by that process.
Great, thank you for that answer. So I think the next question probably goes to Guangwei. The question is, when protein is extracted from almonds, as Juliana just mentioned, we do see lipid as a byproduct. Would you be able to shed some light on how this lipid byproduct might be used?
You know, as you heard from Dr. Baer's presentation, when they're trying to extract the protein, they have to ideally should separate oil first. So oil being separated out, they create the other three stream of product, skim, cream, and then insoluble dietary fiber stuff. The oil is really, you know, almond contain very high high level of vitamin E, and then phytosterol, plant sterols. So those is you know, you remove the oil out, or the almond oil, the fat profile, fatty acid profile is perfect for the just very comparable to the olive oil. So those olive, you know, basically pure almond oil can be used for, you know, for the cooking oil or for salad oil, just a really functional vitamin E rich oil. So this probably have a better use, or can be also used for cosmetics or, you know, or pharmaceutical use. This probably going to be, you're going to see lots of use since there's a demand for the defatted meal use. That means the industry going to produce the good quality cold pressed oil. So those, we will see more uses, but I'm sure that the manufacturers exploring those potential applications.
And that's great. It sounds like there are multiple uses for this byproduct, and you know, all of the other potential uses could also probably be explored going further. So thank you for that. Christine, thank you for those really, really fun demos that you did. They look like delicious recipes, the smoothie and the lemon bar. I would have loved to actually have this webinar, and well, not have a webinar, but a seminar in person so I could have tasted them. But very quickly, since you work with so many proteins, what do you see as the next trend in protein?
Oh, wow. Yes, thanks for the question. And we'll definitely get you some bars. Yeah, in terms of next trends for proteins, I definitely see a deeper exploration of a combination of plant-based proteins. So, you know, we kicked it off. I think there's been so much amazing product development and menu development when it comes to plant-based proteins. I really see us just getting into it a lot deeper. So just looking at different combinations as it pertains to complementary based proteins. So, you know, almonds and pulses being a great combination. Looking at plant-based analogs, I see that expanding quite a bit more, you know, vegan, plant-based, and then also blending and extending with animal-based proteins. There's just a lot of applications, and really, really excited to see where we're going to be taking this in the industry.
Great, thank you very much. Another question I have here is, which is more important, protein quality or protein quantity? So yes, and I realize that I touched upon that during the webinar where I did talk about both. So that's a great question, and I think I can take that one. Both protein quality and quantity are actually important aspects of protein intake. So I wouldn't really play down either. But I would say that in a more plant forward / sustainable dietary pattern world, protein quality can be achieved by ensuring different sources of plant protein in your diet. And this is the concept of complementary proteins presented earlier in the webinar. But this is also something we can do on a daily basis. We do it on a daily basis because none of us is really consuming a single source of any nutrient. So when it comes to protein quantity, however, well, that can actually vary by requirement, which in turn might be dictated by a lot of different factors. So those could be, well, are you a growing child? Or are you an expecting or lactating mother? Are you fighting a devastating illness? Or are you an athlete or just a regular everyday protein consumer like me? So whatever that situation might be, it is important to meet the required protein quantity, and consuming almonds is certainly a healthy and plant-forward way of doing so. So I hope I've answered that question. With that, we have a question for Guangwei. Guangwei, what would you say is the shelf life of almond protein powder in comparison to some of the other almond products, like maybe ground meal or butter?
Well, that's a good question. As I mentioned, existing commercial core almond protein powder is really just a defatted almond meal, right? It's just in protein enhanced almond flour, so it's in different level of protein. So this is probably, since this is a new product, we haven't done, shelf life evaluation for this manufactured product. I'm sure that the manufacturers of the product, they have the information for the shelf life potential of this product. But again, based on my knowledge or understanding how the defatted meal been made, the composition of a defatted meal, I can see the shelf life potential of this defatted meal or almond protein powder should have a comparable shelf life span compared to other manufactured almond form. And then again, shelf life is already, affected by many factors, the packaging and the storage condition. So, almond defatted meal reduce the fat content, but at the same time, because the grinding grinding to the fine powder, so increase the air interaction exposure area. So those is both together probably work against each other. So, but with the proper handling, with the proper package, especially remove the oxygen, stay away from light exposure, those shelf life should have more than 6 months or up to 1 year shelf life, like most of our manufacturer forms. I don't have a solid answer, but I think I get I explain that way that those points can affect the shelf life. I do feel comfortable. We should have a good shelf life if handled properly. Yeah.
Okay, thank you. And can that shelf life be extended if it is stored under certain conditions? And if so, can you shed some light on what conditions should it be stored under to actually help extend that shelf life?
So basically, as our typical recommendation for our other almond forms, store under cold storage, pack it under vacuum or nitrogen flash packaging, and then especially, and for this fine powder, I recommend is trying to package in a dark package, like aluminum pouches, or prevent the light exposure. Don't use those transparent film, plastic film.
Okay.
Those could extend the shelf life. Yeah.
Absolutely. Thank you so much, Guangwei. One last question for Christine here, because I think everyone's really interested in knowing holidays are coming up. And during your demo, Christine, you mentioned a little bit about how almonds, the protein part or almonds themselves can be used for baking. Do you have a couple of, or maybe one quick recipe or tips that you might be able to provide?
Ooh, there's a lot. Yeah, so in terms of baking, there's a lot of different options. So if you want to look at, breakfast items, higher protein based pancakes are always wonderful in the morning. You can incorporate it into bread pudding, into cookies, shortbread cookies, probably some of my favorites. Yeah, there's a lot of different options, which is exciting.
Great. Wow. I think you just made me really, really hungry.
Or lattes with almond milk. It's just kind of nice kickoff in the morning.
Great. That sounds all very, very delicious. Well, I think that's all the questions we have from the audience today. So with that, I'd really like to thank the panel for a very, very informative webinar today. It was a great discussion on almond protein. And we would also like to thank you all for attending and engaging in our conversation. With that, thank you all. And for more information, please visit almonds.com. Thank you and have a great day. Take care. Bye-bye.
Thank you.
Right.
Bye.
Thank you.